I used a little jar, doesn't matter what you use, and I'm actually going to pour mine into a paper cup to make it a little bit easier to pour into my test tube because the test tube is kind of narrow at the top. I'm going to start with the dissolved oxygen because that one takes a little while. Okay, and I'm going to take that tiny little tube and I'm going to fill it all the way to the very, 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 very tippy top. Literally like it's going to overflow, okay? And then find the little silver packet that says DO on the back of it. That stands for dissolved oxygen, okay? And as we learned, dissolved oxygen is very important to our little fishes. If the little fishes and aquatic organisms don't have oxygen in their water, they aren't going to survive, okay? So you're going to take 2 of those little tablets and you're going to put them in the top of your water. And yes, it's going to overflow, so expect to get wet here, okay? And put that lid on nice and tight, okay, so you can get it off. And then you're going to shake it. And you're going to have to shake it for a while because you want those 2 tablets in there to dissolve, okay? So you're going to have to shake it around, da-da-da. Okay? So once it's all shaken up and those 2 tablets have dissolved, you're going to set that aside for about 5 or 10 minutes. It's going to take a little while for that to work, so just set that aside for now. Okay? All right, the next ones, in these tubes, you're going to do the pH, the phosphate, and the nitrate, okay? Unless you have these little strips, and I'll show you what to do with the little strip in just a second. Okay, so the nitrate, the phosphate, and the pH get done in this little tube. So take your little top off, okay, and you're going to take your same water sample, okay, and you're going to fill this up to the 5 milliliter mark, which is about halfway up the tube, okay? See that? Right up there, 5 milliliter mark, right there, okay? All right, so now you've got that filled, and now you're going to take another silver package, and on the back, it should say nitrate, it should say phos, P-H-O-S, or P-H-W-R. P-H-W-R is for pH wide range, phos, P-H-O-S is for phosphate, and nitrate for nitrate. Some of you have 2 nitrate tablets, one yellow and one red, okay? Add the red one first and shake, 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 shake till it dissolves. Add the yellow one second, shake, 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 shake till it dissolves. Others have a single nitrate tablet. Okay, so just check carefully. If you have one red and one yellow, you have the double nitrate. Add the red, shake, shake, shake. Add the yellow, shake, 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 until the tablet dissolves. Okay, and that is the case for all 3 of these. Phosphate, okay, phosphate, nitrate, and pH. You're going to do the same thing each time, okay? So I'm just going to demonstrate the tablet and the shake, okay? So this one, tablet goes in, top goes on, shake, 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 shake until the tablet dissolves, okay? And in all 3 cases, you're going to need to wait a couple of minutes. pH goes very quickly, but phosphate and nitrate need about 5 minutes before they develop a color. Okay. Now, in this particular case, I'm adding a tablet that I know for sure is not going to add a color to mine, unfortunately. So I'm going to end up with a blank result. And that's okay. If we end up with a blank result, it is still a result. It's a negative result, and that's okay. That means that we basically have nothing in our tab. Okay. All right, so my tablet's almost dissolved. Shake, 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 shake. And definitely shake. Don't just invert, okay? This is shaking. Shake, 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 shake. Make sure it's really getting shook up in there. And it's okay if it forms some bubbles. That's not a big deal on this one. You don't want any bubbles in your dissolved oxygen. Your dissolved oxygen is, you sealed it up without any air, and that's important because you don't want to introduce air to the dissolved oxygen one, okay? Where's my camera? There it is. Okay, so there's my dissolved oxygen. My tablet is not quite dissolved, but almost. So let's see if I can get it to finish up. Okay, so now my tablet is dissolved. Okay, see? All dissolved, no tablet. Okay, so I'm going to let that sit for 10 minutes now. Okay, let it sit over there for 10 minutes, okay? Now this one's all dissolved, okay? So let's say this was the pH one, which I said will happen very quickly. What you want to do is you want to take your test tube and you want to put it directly on the card, on the white part of the card, and compare the color of the test tube to the card. Okay, let's try it over here for, let's see, nitrate or phosphate. Okay, so here we go. Let's see, I can line this up better. All right, there we go. All right, so say that I was measuring nitrate with this particular one. Okay, nitrate comes out as a pink color, but there's no hint of pink in this at all, is there? Even with my pink shirt on. Probably a bad idea. Shouldn't have worn pink. But pink, if this had any nitrate in it, it would show up as some shade of pink. You can't interpolate between the colors. You can only say it's closest to. So it's either closer to 5 parts per million, 20 parts per million, or 40 parts per million. In this case, I would simply put 0 because I have no color whatsoever. Okay. Now, if I was looking at phosphate over here, it's kind of hard to get used to this camera being backwards from me. Okay. All right. So if I was phosphate, I'd be looking for a blue color, 1 part per million, 2 parts per million, or 4 parts per million. And again, I cannot interpolate. I have to say it's closest to one or the other, okay? Or it's between this and this. So this one here, again, if it's clear, your parts per million are going to be 0, okay? So you may end up with a 0 on phosphate and nitrate. You may even end up with a 0 on dissolved oxygen, although I hope not. pH, on the other hand, on the other side, you should end up with some color, but I wasn't testing for pH in this one, so I didn't end up with any color, okay? Now, turbidity is kind of easy, but I'm going to change my water, okay, because I don't want the interference of whatever the chemical was that I put in there. So I'm going to dump that out, and I'm going to add some, a new sample, okay? Same water sample, just a new clean water sample, okay? And I'm going to put it in that tube, and then I'm going to hold the tube directly over those little circles, okay? Whoop. Okay, so which one does it look closest to, okay? Is it clear? I can see the black very clearly, okay? Or is it gray? Or is it really gray? So in this case, it looks to me, see if I can get this lined up better. This is hard to do backwards. Not used to this. Okay, so if I look at this, all right, there's a little bit of cloudiness going on there, right? Okay, so let's put this like that. Here we go. Okay, so which one does it look like? Does it look like the black is pure black? Oh, goodness. It's hard to do backwards. I just don't get that. There we go. All right, so is it pure black? Is it a little bit gray? Or is it dark gray? Okay, I think I'm pretty close to 0. I don't really see any difference in the color of the black between this. So that would say the turbidity or the suspended solids in my water was 0 or pretty close to 0. Okay, so that's how we read turbidity off of here. Okay, we didn't do coliform, so don't worry about that one. You're going to do pH. You're going to do phosphate over here, and you're going to do nitrate here. Okay, now for those of you that did not get the tablets for something, and I believe it's pH that you didn't get tablets for, because excuse me, phosphate, because I was really low on phosphates. You have this little strip, okay? You see these little colored squares on it. Okay, what you're going to do is basically just dip it into your water and get those little squares wet. Doesn't have to stay in there for very long. They just have to get the little squares, all the little squares wet. So get the little squares wet, and then you're going to use the bottle to match it up, okay? And you see there. So you see how my colors are changing, and I'm going to try to match up my colors, okay? So I try to match those colors up with the ones at the end, okay? So for those of you that are doing the strips, okay, that's what you do with those. Everybody else is going to have the tablets. And I believe it's only one of you that has the strip. And if it doesn't work out, don't stress it. If if the phosphate doesn't come out, then we have a null data set, and that's okay. Sometimes we aren't going to get a good reading for some reason. In some cases, it might be because the tablets are old and they're not working right. Sometimes it's simply because there's just no phosphate in that particular sample. Okay. All right, so here's my dissolved oxygen. Hasn't been quite 10 minutes yet, but you can see that I'm getting a little bit of color. It's no longer pure white, is it? Okay, so go over here. Okay, I'm getting a little bit of color, but I haven't let this sit long enough yet. It's going to have to be a few more minutes before I can read that one. But again, you're going to take the bottle, put it on the white portion, and then try to read what color pink it comes out. And again, this one should come out pretty good. I'm hoping anyway, because this was my pond, and if my pond doesn't have any water, any oxygen in it, then my fish are going to die, which would not be a good thing. Okay, all right, so water sample. Okay, you got your water sample. You see mine is very green from my pond. It's got some floaty stuff in it. Okay, you got your test tube. Fill your test tube up for the nitrate, phosphate, and pH. Fill it up to the 5 milliliter mark. Use the tablets. Remember, dissolved oxygen gets 2. Nitrate gets 2 if you have red and yellow nitrates. If you have a single nitrate that's black, it's just 1 tablet. Okay. Phosphate gets 1 tablet, and pH gets 1 tablet. Okay. If you don't have tablets and you have the strips, you simply get the strips wet and then compare them to the comparator. And hopefully I remembered to give you that. And if I didn't, let me know and I'll send it to you. Okay? All right. So if you have any questions about the water quality, please don't hesitate to ask. If you're in the middle of doing it, text me rather than email me, okay? Because once you've used the tablets, that's it. There isn't anything left, okay? And you can't let them sit for a long period of time before you read them, okay? No more than 5 or 10 minutes. The longer you leave, especially phosphate, the longer you leave it, the more it reacts, and it's not giving you an actual reading. So if you have a problem while you are doing the lab, please just text me, and I'm usually around my phone, and I'm usually in a position where I can text back pretty quickly, unless I'm driving. No texting and driving. So let me know if you have a question during the lab via text, okay? And remember, you need 3 water samples, okay? 1 tap water, okay? 1 sort of stream or river, doesn't have to be anything fancy, can just be a little runoff stream in your backyard. And some sort of standing water. And honestly, that could even just be a puddle after rain, okay? But you need some kind of a standing water sample, a running water sample, and then a tap water sample, okay? After you have used this test tube for each test, rinse it out, if you can, with some bottled water, okay? Not your tap water, because your tap water contains chlorine and it'll change the reaction. So if you have some bottled water, rinse it out with some bottled water between each test so that you don't contaminate from one to the other. Okay, I know that's a little tedious, but I just didn't have enough test tubes to send you 9 test tubes for every test, okay? So just rinse it out with some bottled water and then do the next test. Try to do either all of the same water sample, finish 1 water sample all the way through, and then switch to the next water sample all the way through, and then the 3rd water sample all the way through, rather than trying to do the same test for each water sample. That way you know that the test tube is not contaminated with the different water samples between tests. Okay, does that make sense? Okay, let me just take one more quick look at my dissolved oxygen. Oh, look there, yep, I'm getting a little bit of pink. Okay, so you can see, let me see if I can get it up there close enough. You can see there's just a little tint of pink in mine. Okay, where's my dissolved oxygen? Over here, there we go. Little bit of pink. Okay, let's see. Here we go. All right, so I'm definitely not 0 because I've got a little bit of pink in there. So I'm somewhere between 0 and 4, but I'm definitely not an 8. Okay, so That's what we want to read, okay? Again, any questions while you're doing the lab, text me 336-392-9425, okay? And just in the text, tell me who you are. So when I look at your phone number, I don't go, who is this? Okay, so let me know who you are, and good luck with the water quality tests.